Friday, finally. 3-day weekend. You guys going to miss biochemistry on Monday? No? I won't either. How's that? But I'll miss you guys. How's that? All right. So I got started last time talking about mitochondria, and I want to go through mitochondria, and we'll get into oxidative phosphorylation today. And as we get into oxidative phosphorylation, we begin to realize that the central sort of control area for a great deal of metabolism actually arises as a result of controls in the mitochondria. And the controls, as I've told you so far, are very simple. It's really the level of NAD and FAD. That really is what it comes down to. And the levels of NAD and FAD are, in turn, dependent upon the amount of oxygen available. Now, when we get to oxidative phosphorylation, we'll see one more thing that will affect the overall regulation, and that's ATP/ADP. And it's a combination of those four things, FAD, NAD, oxygen, and ATP/NAD, ATP/ADP, that really govern virtually all of our energetic metabolism. Those things right there. So you understand those and how they affect that, you will understand energetic metabolism considerably. Okay, well, this figure shows that same thing that I showed you last time in terms of the flow of electrons in a little bit different way. And it's showing it with respect to free energy on the y-axis. So we see that free energy is decreasing, meaning this is energetically favorable for the movement of electrons going through the electron transport system. All right? This figure shows the complex 1, complex 3, and complex 4. And it shows the intermediaries, which are, in this case, Coenzyme Q and cytochrome C over here. Okay? And we also see that complex 2 electrons feed into the Coenzyme Q as well. So we remember that movement of electrons through the complexes 1, 3, and 4 result in the pumping of protons outside of the mitochondrion. Now, one of your tendencies is going to be to really try to count things. How many protons per ATP, et cetera, et cetera. This is an area of biochemistry that people have worked on for years, and people get into literally fist fights about how many it really corresponds to, and it really doesn't matter. It really doesn't matter for our purposes. What we will care about is the fact that protons get pumped. And it's the pumping of those protons that ultimately will result in the synthesis of ATP. So we're not going to try to count protons. It's just not going to be worthwhile. But we will note that protons protons pumped as a result of electrons from NADH will be greater than the number of protons pumped as a result of electrons coming from FADH2. Now you'll notice I'm saying protons and electrons, and I actually stumbled over it myself. It's important for you to not mix up protons and electrons. This is an electron transport system, and an electron transport system pumps protons. All right? So don't mix those up. It's not a proton transport system. It's not electrons that get pumped. Okay? So just keep those things in mind. All right. Well, ultimately, of course, the terminal electron acceptor is oxygen, and oxygen accepts electrons and picks up a couple protons from the water and from the solution and becomes water. Okay. So one of the ways people have understood and studied the electron transport system is by studying things that inhibit the transport of electrons in the electron transport system. Okay? Now, these names that you see here, NADH/Q reductase, that one corresponds to complex 1. All right? And when we see Q cytochrome C oxidative reductase, that corresponds to complex 3. And when we see cytochrome C oxidase, that corresponds to complex 4. All right? When we look at the movement of electrons, we see that certain compounds inhibit the movement of electrons through the complexes, and they're known as poisons. All right? So up here, rotenone and amytal both inhibit the movement of electrons through complex 1. Rotenone is a plant compound. It's actually made by plants as a defense mechanism. And it is fairly fatal to insects that consume it. And people actually use rotenone as an organic insecticide because it's a plant product, and they put it onto plants like tomato plants, for example, and keep insects from infecting them. It works by inhibiting this oxidoreductase, by inhibiting the complex 1 movement of electrons. Well, it's not quite as poisonous to us, all right? And that's fortunate, although I wouldn't recommend you go out and make a diet of rotenone because it probably wouldn't be a good idea. But the point is that electrons have other ways of moving through the system, okay, that bypass complex 1. So, for example, if we were to bring electrons in from FADH2, it would come in through complex 2 and would bypass this system and come in down here, and so we would still have some kind of an electron transport system going on. We see that movement of electrons through complex 3 is blocked by Antimycin A. Okay? Now, what does this mean if I say it's blocked? Well, it means that if I add one of these compounds, let's say I added some Antimycin A, electrons would flow down to here, and they would stop. And when they stopped here, the system would back up to here. It would back up to here. And basically, everything that would be trying to donate electrons, NADH and FADH2, wouldn't be able to do it. And so the reason that they're poisonous is, of course, when you can't make NAD and you can't make FAD, you're going to stop the citric acid cycle. You're going to stop glycolysis ultimately. Yes? Antimycin is not accepting electrons. It's blocking the movement of electrons. It's not accepting them. Okay? So it's physically stopping the movement of electrons through the complex 3. So once it's there, it's blocked it. Okay? Okay. Now, if we look at the bottom, we see 3 things block the movement of electrons through complex 4. All right? These compounds turn out to be the most poisonous because there's no way of getting around them. You can actually get around Antimycin 2 with some unusual chemistry, but you can't get around complex 4 because it's the terminal electron acceptor. And look what inhibits complex 4. Cyanide. That's azide. And then, here's our friend, carbon monoxide. When I told you before that carbon monoxide kills you in 2 ways, one by binding to your hemoglobin, this is the second way that carbon monoxide kills you. It blocks the movement of electrons through complex 4 and shuts down the electron transport system, period. Well, if you shut down the electron transport system, what's going to happen to your consumption of oxygen? Are you going to need oxygen? You really don't need oxygen, right? So what this system is doing is it's helping to suffocate you. Right? It's helping to suffocate you. Okay. Now, this block of complex 4 is, as I said, very, very nasty. It's very fatal. And it's not something that we want to mess with, certainly. But in the laboratory, we use these as tools to study the movement of electron transport. And people who worry about how many protons get pumped out and so on and so forth, and all that adding up, use these inhibitors as ways of studying that. So they have some very useful research purposes. Yes, sir?
Could you please explain again why we don't need oxygen anymore in this case?
Can I explain why you don't need oxygen anymore? There's no electrons coming through. There's no place to put electrons. So if you have no electrons coming through, you're not going to use any oxygen at all. Right? Okay? So you're not going to use a drop of oxygen if you block electron transport. So if I told you on an exam, for example, that a person took cyanide and then started breathing very heavily, okay, would that make any sense? It probably wouldn't, right? Because they're not using any oxygen. Not using any oxygen at all. Okay. So these are the kinds of things we think about with respiratory control. I'll give you a lot more examples with those as we go along. Yes? So his question is, will the body think that it needs more oxygen because it can't use oxygen? And I'm not a physician, but my strong impression is the body would just think, you know, I got plenty of oxygen. When you're sitting here, you're not breathing heavily because you're not using very much oxygen. So if you're using less oxygen, you would probably breathe less heavily than you are right now, right? Yes?
Cyanide kills very fast, and if you don't use oxygen, I'm wondering why would people die very fast?
Why do people die very fast? Because you can't go very long without having your citric acid cycle working.
Also, it's really, really short period of time.
Yeah, it doesn't take very long. Well, think about it, okay? Citric acid cycle, the oxidation of glucose gives us 38 ATPs. No citric acid cycle, the oxidation of glucose gives us 2. Okay? We can't sustain that for very long. Not across our whole body. I'm sorry? I'm not talking about seconds. I'm not going to do the experiment to find out how long it would kill me. But, but you might want to try this. I don't know on me. But it's going to kill you fairly quickly. It's going to kill you within minutes. Yeah. Okay. Let's see. Quinones. So coenzyme Q is in a category of molecule known as quinones. And somebody asked me yesterday about NADH and said, well, on Wednesday, I guess, about NADH. And they saw that the H went on, but they didn't understand the electrons going on. I said, well, look at the structures. You'll see that the actual ring configurations for electrons actually changes. And quinones also have changes in their electronic structure that happens, and that's how they accommodate the electrons that they're accepting. Here's the oxidized form of coenzyme Q. Okay? Here is coenzyme Q that has accepted 1 electron and 1 proton. All right? Here is coenzyme Q that has accepted 2 electrons and 2 protons. And you can see that we're going from 2 double bonds over here, 2 oxygens, to 3 double bonds, 1 oxygen, and an oxygen with an unpaired electron. Over here, 3 double bonds and 2 hydroxides. Okay? So there are electronic changes that are happening, and they have to because this guy is accepting electrons. Okay. Well, so that's why coenzyme Q that I said was a traffic cop. It's able to accept 2 and pass them off 1 at a time. We're going to see how that happens in something called the Q cycle. Before I show you the Q cycle, I just want to show you some of these other molecules. Okay. The complex 1 is a complex that moves electrons mostly through it. Remember, it's a complex of many proteins, but there are molecules in there called flavins. And flavins are other carriers of electrons. FAD, flavin adenine dinucleotide, is a flavin. Okay? So complex 1 and complex 2 have flavins. Here's one called flavin mononucleotide. Okay? So complex 1s are full of flavins. Complex 1 also contains what are called iron-sulfur complexes. And iron-sulfur complexes can come in quite a wide variety of forms. You can see some variety up here. And we remember that iron can exist in 2 different oxidative states. Iron can exist in the +2 form, which is the reduced form, the relatively reduced form, and the +3 form, which is the relatively oxidized form. That's a difference of 1 electron. Okay? So we can see, really, that these iron centers can participate in the grabbing and passing of electrons as they're moving through this overall cycle. So iron-sulfur complexes, which are part of what we call iron-sulfur proteins, are very important in electron transfer. Here's a schematic diagram that shows now the movement of electrons. There's some iron-sulfur proteins. There's flavin mononucleotide. I'm not going to ask you to draw this, so don't worry about that. But I just show you this to illustrate that this is a complex of these many different electron-carrying molecules. The aim is really doing 2 things, or 3 things. 1, producing NAD. 2, pumping protons. And 3, passing those electrons off to coenzyme Q. So complex 1 accepts a pair of electrons. It passes that pair of electrons off to coenzyme Q. And so we designate coenzyme Q by the term Q when it's fully oxidized and QH2 when it's fully reduced. Q when it's fully oxidized and QH2 when it's fully reduced. Okay. So now let's tackle something called the Q cycle. The Q cycle happens at complex 3. The Q cycle happens at complex 3. All right? Now, what's happening here in the Q cycle? All right? Well, here's something called the Q pool. And students always get confused, but what's the Q pool? What's the Q pool? The Q pool is the sum of all of the coenzyme Q that's in the intermitochondrial membrane. Just like a swimming pool is a collection of all the water that's laying in that thing that we call a pool. Okay? It's just a mixture of all those things. So the Q pool is going to have millions of coenzyme Qs out here, some of which are going to be QH2, meaning they're going to be fully reduced, and some of them which are going to have Q, which are going to be fully oxidized. Okay? So that's what the Q pool is going to consist of. Right? So we have both of those out there in the intermitochondrial membrane. The Q pool donates to complex 3 to start the cycle. It donates a reduced coenzyme Q. That's the QH2. So the QH2 has come in from the Q pool. It has entered complex 3. And when it enters complex 3, it has 2 electrons and 2 protons to donate. It does that. It donates 1 proton to a Q that is oxidized that was sitting in the complex. So the complex started with an oxidized Q sitting in the bottom of it right here. That oxidized Q gains an electron. That electron, or that one is shown as Q dot minus, meaning it's gained an electron. Well, QH2 came in with 2 electrons and 2 protons. What happens to the other electron and the other 2 protons? The other electron is donated to cytochrome C. So cytochrome C gains the other electron, and now the 2 protons are released. And the 2 protons right here are pumped out. That is, they're put out of the mitochondrial inner membrane, and you've seen some pumping occurring right here. Okay? At that point, we're left with Q. Because Q came in with QH2, it's donated 2 protons, it's donated 2 electrons, Q is left. So what happens to Q? It goes back out into the Q pool. Okay? So we've just done the first half of the cycle. Coenzyme Q came in with 2 electrons, 2 protons, 1 electron to coenzyme Q, 1 electron to cytochrome C, 1 electron to coenzyme Q, 2 protons pumped, Q exits complex 3. Okay? With me? Second half of the cycle sounds like something completely different, and it's exactly the same thing that we saw before. A QH2 comes in from the pool, and 1 electron goes on to Q minus. It picks up 2 protons and becomes reduced coenzyme Q, which now gets kicked out into the Q pool. The other electron goes to cytochrome C, and the 2 protons are pumped out. We've taken in 2 QH2s. We've released 1, so we've had a net use of 1 QH2. Yes? What's the purpose of giving an electron to the Q right here, right? Okay. Well, this Q can't come out until it's either a Q or a QH2. The Q pool can't have a single electron. So it has to donate it to something, and it donates it to this Q, which in this complex can hold a single electron, but it can't go out in the Q pool like that. It stays there. So this guy's just sitting here with 1 electron, waiting for the second electron to come. Yes? Same complex, that's right. What's that? This is the same complex, they're both complex 3, that's correct. Okay, so 2 QH2s come in. Both of them split their electrons, 1 to cytochrome C, 1 to coenzyme Q. All right? Both cases, 2 protons get pumped out, and we're done. We've done the Q cycle. So the purpose of the cycle is to participate in the electron transport system. So we're seeing now the movement of electrons. Remember, we started with electrons coming in from coenzyme Q. They've passed to complex 3, and now complex 3 is passing them off to cytochrome C. So this is electron transport up close and personal. That's you're actually seeing this happening right here at this complex. Okay? That's the purpose. The purpose is to participate in the electron transport system and to pump the protons, because we see the protons getting pumped out as well. Yes? So the two protons that come from here come from the mitochondrial matrix. That's correct. So this is the mitochondrial matrix down here. This is the intermembrane space up here. Yes? Yep. That's right. So her question is, the proteins that are sitting there in complex 3, they're just a guide for this process. We can think of complex 3 as a docking station. That's the way I think about it. Right? And remember, these are big honking complexes. So that's why we need these little guys like coenzyme Q and cytochrome C to ferry the electrons between the complexes. So the complexes are, in fact, kind of like docking stations. All right. Clear as mud? You guys are dead today. Yes? His question is, why does QH2 split off both electrons? That's similar to her question over here. Why doesn't it split off one? Because it can't come out with a single electron. It will not exist freely in the membrane with a single electron. The only place it exists freely with a single electron is protected in this complex. This is actually a radical right here. All right? Meaning it's fairly reactive. This complex is set up to keep that radical from reacting with something. Okay? That's the reason why. All right. Okay, let's turn our attention to complex 4. Complex 4 is pretty cool. All right? Starting with complex 3, we got another group of electron carriers that started to participate. In complex 3, we start to see specialized proteins called cytochromes. Cytochrome C, which you see as that little farrier between 3 and 4, is one of those cytochromes, but there's also cytochromes within complex 3, and there are also cytochromes within complex 4. These are proteins that have heme groups. So cytochromes have heme groups, just like hemoglobin has heme groups. Okay? And heme groups, you remember, have iron at their center, in the case of hemoglobin, for carrying oxygen. They're not carrying oxygen, at least here, but that's what they're doing in hemoglobin. All right? When we get to complex 4, the heme groups are also there, but one of the heme groups, instead of having iron, has copper. Now, these irons that we see in the heme groups and the copper that we see in the heme groups, all right, they all have two different electronic configurations. Iron can be +2, +3, meaning a difference of 1 electron. Copper can be +1, +2, a difference of 1 electron. So again, we see the ability to grab an electron and let it go, and grab an electron and let it go. So this different oxidation states of the metals that we see, very important for the movement of electrons. This figure shows the attachment of a copper to a histidine, okay, inside of complex 4. Okay, here's the heme, just to remind you what that looks like. If we replace that iron with a copper, then we have one of the hemes in complex 4. Okay. Here's another mechanism, and I think this one's probably a little bit easier to understand. It looks a little bit more complicated, but it's actually not too bad. All right. Here is complex 4, the thing in the rectangle. All right. Complex 4 is, again, we can think about it as a docking station. Complex 4 has an environment that is stabilizing some of the radicals that we see coming from the movement of electrons. Now remember that at this point we're moving electrons individually. They're coming in one by one from cytochrome C. Cytochrome C is coming up here. Okay? When cytochrome C comes in, all right, we start with this overall complex, all right, looking something like it looks right here. Okay? So here's here's the complex, and it's sort of depicting for you where the electrons are going to move through. Okay? So we're starting out with the addition of 2 electrons, and the addition of 2 electrons causes these guys to be full of electrons. That's what's showing here in the red. So the iron, we have an iron in the heme, we have a copper in the heme, and we have the these guys have electrons, and an oxygen then comes in. Remember, oxygen is going to be the terminal electron carrier. Oxygen comes in, and it sees these electrons, and it binds, making a peroxide bridge like we see here. And that peroxide bridge is taking those electrons away because oxygen is very electronegative and grabs electrons for itself. Okay? Next step of the process, 2 more cytochrome C. By the way, we had 2 to start the process. We have 2 more here. All right? 2 more cytochrome C come in. And what they do then is they take that peroxide and they reduce it to 2 hydroxyls. So we have 2 hydroxyls, where up here we have molecular oxygen. So we've used 4 electrons, 2 electrons here and 2 electrons here, to convert those 2 oxygens into 2 hydroxides. At that point, all we have to do is add 2 protons to peel off those hydroxides and leave behind oxidized iron and copper. That produces 2 H2Os.
Question.
Yes?
Are these electrons coming in by 2H2?
No, they're coming in by cytochrome C. Cytochrome C. Okay? Remember, the Q is only coming into complex 3. So what comes into complex 4 is cytochrome C. So these are coming in. There takes 4 of them to do this. So it takes 4 electrons to reduce 1 molecular oxygen into 2 molecules of water. Get that down, right? 4 electrons reduce 1 molecular oxygen into 2 molecular waters. Yes, sir?
So each cytochrome C carries 1.
Each cytochrome C carries 1 electron. Everything after complex 3 moves in 1 electron units. Yes? Does it matter if the copper gets the electron first or the iron gets the electron first? It probably does, but for our purposes, it doesn't. Yes? What are the source of the protons and electrons? Well, the source of the electrons, of course, is cytochrome C. The ultimate source of the electrons was NADH or FADH2. And the source of the protons is the protons are coming, as you see, out of the solution. So any solution, if you have a pH of 7, right, neutral water, pH 7, that means it has 10 to the minus 7th molar protons. So you've got protons in water anytime you've got water. You can't escape that. So there's plenty of protons for water to donate to this process. So that's where those protons are coming from. Okay? Very good. Good questions. That answer your question, I hope? Okay. Anybody else? Okay. So there is what's happening. So now you can see why oxygen is needed. You can also see something that leads us to our next discussion, which is that what happens if this process gets interrupted? What happens if it doesn't complete properly? Well, if it doesn't complete properly, what we actually do is we end up creating molecules that have an extra electron or need an electron or whatever. There's something that's not good there. And unpaired electrons are really problematic because molecules that have unpaired electrons are very, very reactive. Okay? Very reactive. One of the things, I think I mentioned this earlier in the class, one of the things we see with mitochondria is if we look at mitochondria that are very old, from a cell that's very old, like my cells compared to your cells, the mitochondria look real beat up. And the reason that they look real beat up is because over time, okay, there have been the release of reactive oxygen species, what we call them, that's things with unpaired electrons, and those things with unpaired electrons have reacted with proteins and other things in the membranes of the mitochondria. Your mitochondria get damaged with age. One of the strategies for maintaining youth is to try to maintain the integrity of mitochondria. Okay? And there's some very interesting things out there, very interesting strategies that people are using to do that. Okay. All right. Let's see. That's the overall process. We don't need to worry too much about that. Okay. Well, the reactive oxygen species that I talked about leads us to a very interesting enzyme called superoxide dismutase. Superoxide dismutase is an enzyme that's a very important enzyme. We find it in virtually every living cell. That tells us it's very important. It tells us that, and what this enzyme does is it catalyzes the conversion of something called superoxide, which is what this guy is here, oxygen with an extra electron. Superoxide gets converted back to molecular oxygen. It also gets converted to hydrogen peroxide. Now, hydrogen peroxide is a fairly reactive molecule as well, but it's not nearly as reactive as superoxide is. This enzyme uses the ping-pong mechanism that we talked about last term. Anybody remember the ping-pong mechanism from last term? Back and forth, that's right. The enzyme bounces back and forth between two different states. And we can see that here in color. Here's the superoxide, that's the M. Superoxide, in the oxidized form, okay, is able to take an electron. And look who has an extra electron. Superoxide. It takes the electron away from superoxide and produces molecular oxygen. That leaves the superoxide dismutase in the reduced form. So superoxide dismutase goes from being in the oxidized form to being in the reduced form. Meanwhile, superoxide is converted into molecular oxygen. Okay, I'll repeat that. Superoxide dismutase starts in the oxidized form and goes to the reduced form. And it does that by picking up an electron from the molecule called superoxide to make molecular oxygen. Yes? Okay. So you didn't see a superoxide before. Okay? But I said that the process before can leave things with unpaired electrons. Unpaired electrons can generate superoxides. Okay? So I haven't shown you how you do that, but I'm just saying if that process gets screwed up and we don't make two waters, then we are ultimately going to make superoxides. Mm-hmm. Yep. Okay? Now, here's the reduced superoxide dismutase. We've got to get it back to the oxidized form, so that's what happens down here below. Superoxide dismutase in the reduced form has an extra electron. It encounters a superoxide, and what it does is it converts the superoxide into hydrogen peroxide. That leaves the superoxide dismutase back in the oxidized form. And we're ready to start the process again. So we've gone ping, pong, right? All right. So this enzyme is really interesting. I'd say it's really important. We know, for example, that in the case, there's Dr. Joe Beckman here at Oregon State is a world expert on studying superoxide dismutase. And the reason he studies it is because it's involved in Lou Gehrig's disease, amyotrophic lateral sclerosis. And some very interesting ideas he has about how that enzyme plays a role in the progression of the disease. I saw a hand back here. Question? What effect does superoxide have on what? On the body. Well, on anything, superoxide is extraordinarily reactive. So as I said in the class before, cells are control freaks. They want to control the reactions happening inside of them, and that's why they have enzymes. Things that are reactive without enzymes are problems for the cell because they will react indiscriminately. So they'll react with membranes. They'll react with proteins. They'll react with metabolites. They'll react with DNA. They'll react with the first thing that they happen to bump into. So the damage that they cause is extensive. Okay? And cells try to control that by having this enzyme. And our cells make a fair amount of this enzyme. Yes?
So what happens to the hydrogen peroxide? Does it also
What happens to the hydrogen peroxide? Very good question. This guy is fairly reactive also and can be problematic for cells because it's also fairly reactive, but it's not as reactive as superoxide dismutase is. Okay? We have an enzyme in our body called catalase, C-A-T-A-L-A-S-E, that breaks down hydrogen peroxide. Okay? So we don't want too much of this going around. Turns out hydrogen peroxide is kind of an interesting molecule, though, because cells actually use it for intracellular signaling. That's kind of cool, but we won't talk about that here. Okay, so that is, one last thing I'll talk about electron transport, and that is cytochrome C. Cytochrome C is a very interesting and important protein. We can find cytochrome C in species that go all the way from bacteria to human beings. And you're probably thinking, well, bacteria don't have mitochondria. Why do they have cytochrome C? And the answer is because they do electron transport in their own membranes, their cell membrane. They don't do it because they don't have mitochondria. They do it in their own cell membranes. So they need cytochrome C, and they have it. When we compare the cytochrome Cs of different species, we see some commonality of structure. Look at the common structures that we see from these from very different species. This coming from tuna, this coming from Rhodospirillum rubrum, and this from Paracoccus, whatever that is, blah. All right? But the point is that they're not that different. The structure is conserved, which tells us that that structure, if we put human beings' cytochrome C up here, it would look very similar as well. When we see structures conserved over evolutionary time, it tells us that the structure is very critical for the function of the cell. Very, very critical. Okay? We use cytochrome C as a way of comparing evolutionary history because even though the structure is conserved, the actual amino acid sequence isn't completely conserved because there's some variability that happens. And so we can compare, for example, the cytochrome C of bacteria with that of human beings and get an entire evolutionary profile from that entire distance. And that's done quite commonly, actually. Okay. So let's turn our attention now to oxidative phosphorylation. We've talked about cytochrome C, and we've talked about, electron transport, and what we've done so far is we've charged the battery. That pumping of the protons out of the mitochondrial intermembrane, out of the matrix into the intermembrane space has caused there to be a proton gradient. And a proton gradient means there's a difference of charge, and a difference of charge means that there's an electrical difference between them. We've literally charged the battery. If we're going to realize something from that charge, we've got to be able to use that charge difference. And the way we do that is with the process of oxidative phosphorylation. That process of oxidative phosphorylation converts electrical charge into ATP. Now that is something that's remarkable. It converts electrical charge into ATP. Human beings don't know how to do that in a laboratory. All right? We know how to do it in a mitochondrion, but we can't construct something that converts electricity into ATP. Okay? To remind you, Peter Mitchell proposed this back in the 60s. It was called the chemiosmotic hypothesis. The chemiosmotic hypothesis had several elements to it. It said, the first thing I told you was that the intermitochondrial membrane had to be intact. Okay? And that's correct. It has to be intact. It tells us that electrons move through complexes. And that turns out to be correct as well. It tells us that the movement of electrons through the complexes causes a proton gradient to be formed. And that's correct. And the last thing it says is that the movement of the protons back into the mitochondrial matrix is the energy source for making ATP. The movement of the protons back into the mitochondrial matrix is the energy source for making the ATP. Okay? We're going to see how that happens. And this is really remarkable. Okay? Here's a schematic of the overall process. Movement of electrons. Okay, so here are the four complexes. Here's the gray area is the inner mitochondrial membrane. We can imagine this goes all the way around, okay, because it has to be intact. Here's the electrons coming I'm sorry, the electrons are actually coming in. Actually, this is a dumb figure. Sorry. Well, it is because they've bent the thing around backwards. This guy here is the matrix on the outside. This is the intramembrane space in here. Yeah, that's why it's confusing. That's why I got confused too. All right. So what's happening is here's the matrix. Here's the intramembrane space. All right. You can see it labeled there as well. All right. Passing of, so electrons are coming here. They're passing through these guys, and we see pumping of protons to the intramembrane space. Proton, proton, proton. We're not counting protons, but those are the protons that are going there. We've got a gradient. Those protons want to come back into the matrix. They pass through something that has several names. The something that has several names, one of the names is right there. It's called ATP synthase. It's also commonly called complex 5. Complex 5. Complex 5, all right, is this remarkable molecular motor that I'm going to show you more about in just a little bit. Okay? This high gradient of protons passes through complex 5, Gesundheit, and causes this little rotor here to spin, and the spinning of that rotor causes ATP to be created. Okay? If I don't have an intact mitochondrial membrane, let's say I've got a hole over here, then what's going to happen to my protons? Well, they're going to go out the hole. They're not going to go through this thing. So that's why the intact mitochondrial inner membrane is absolutely critical. Okay. This is actually what it looks like. Okay? This is actually what it looks like. The protons are moving, in this case, through from here down to here. Okay? The protons are moving. Am I saying that right? Yes, I'm saying that right. Okay. This guy looks like a mushroom. Oh, sorry. This guy looks like a mushroom. All right? It's kind of upside down mushroom right here. There's a stalk to the mushroom, and there's the head to the mushroom. All right? Yes?
I'm just curious. I can understand the biochemistry can identify different units, but how can they actually realize that actually there is spinning? I'm very curious what method would allow you to realize there is actually spinning of such tiny little molecules.
Okay, so his question is, how do we know it's spinning? And I'm going to save that until Wednesday of next week to tell you. But there is a very cool experiment people did to actually demonstrate that it spins. Very cool experiment. Okay? All right. Okay. This is up close and personal, the head of that mushroom. We're looking at the mushroom from the head looking down. Okay? The head looking down. And there is a little subunit. By the way, I don't care that you know the names of these subunits. I'm just showing you this. A little subunit called gamma. And the little subunit called gamma, I can think of as a pointer. It's pointing to something. Okay? I have 3 identical pairs of units, an alpha and a beta, an alpha and a beta, and an alpha and a beta. And you'll notice they're colored a little bit differently, and that's because these guys, depending upon which direction the gamma is pointing, determines a slight change in shape of the units. Here's the beta unit. Okay? It determines a slight change in the shape of the beta unit. Okay? Why is that important? Well, it turns out that the beta unit can exist in 3 different, slightly different forms. One form is called open, one form is called loose, and one form is called tight. All right? What are the functions of those forms? All right? The functions, and they're not shown very well in this figure, the function of the open is basically to open up and release what it made. What did it make? Well, down here it made ATP. So the function of open is to release the ATP. It's not really shown well here. Okay? What's the form of the loose? The form of the loose is to bind ADP and PI. That is inorganic phosphate. All right? What's the function of the T? The function of the T is to squeeze together the ADP and the PI to make ATP. Now, What's happening here? These all exist as that big mushroom structure that you saw. At any given time, there's an O, there's an L, and there's a T. If I start with an O, all right, I have a configuration like I see here. Let's imagine this is my pointer. All right, my little indented pointer there says you are going to be an O. This X-dented pointer here says you're going to be a T. And this lack of pointer over here says you're going to be an L. Okay? That's probably not the right way I wanted to say that, was it? Yes, okay, that's right. That is right. Okay. So that's the way I want it to be. All right? Now, the protons moving through this complex cause this, the subunit, let me show you here. The movement of electrons through the complex cause this little blue ring to spin. It's the blue ring that spins. And the gamma is attached to the blue ring. So the gamma that you just saw is there. All right? So movement of protons is causing the blue ring to spin. So now, when we look at the gamma, we see the gamma spinning. This gamma on this figure is actually going to spin in the counterclockwise direction. It doesn't matter if it's clockwise or counterclockwise. I'm just showing you on here because it depends on whether you're looking at it up or down. All right. So when this guy spins, what's going to happen? The pointer, the X-dented pointer, is going to point over here. What's L going to become? L will become T, right? And that's really good because we've got an ADP and a PI sitting there waiting for it. And what's it going to do? It's going to make ATP, right? All right? Now, this flat thing is going to spin this direction. O is going to become L, which means it's going to accept ADP and PI, right? This indented pointer down is going to point down here, and T is going to become O. And what is O going to do? Release the ATP that it made. So this thing just spins, 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 and every time it spins, it produces an ATP. Question? The mushroom head stays still. It's the gamma that's spinning, and it's spinning because that little blue ring inside of there is spinning. Yes?
Why O, not like a crystal issue, why O has still ATP plus PI?
That figure is just not a good figure. It should show it as empty. Yeah. It should be empty. Okay? Okay, it's a very cool process. Right? Yeah. So say it again. The alpha subunit really isn't playing a role in this process. It's holding on to ATP, but it's not really doing anything here. Yeah. Okay. All right, we're getting there. We're getting there. All right. This is the process. The book's figures are not very good. I kind of like the video, so let's see if we can get the video to work. Oh. Oh. There was a beautiful video that really showed that. I'll see if I can find another video to show you that process. You guys are sounding like you're ready to go. We're not ready to go yet. We have to sing a song, don't we? Oh, I'm a little mitochondrion who gives you energy. I use my proton gradient to make the ATPs. He's a little mitochondrion who gives us energy. He uses proton gradients to make some ATPs. Electrons flow through complex 2 to traffic cop CoQ. Whenever they arrive, they're in an FADH2. Electrons flow through complex 2 to traffic cop CoQ. Whenever they arrive, they're in an FADH2. Yes, tightly coupled is my state unless I get a hole created by my membrane by some dinitrophenol. Yes, tightly coupled is his state unless he gets a hole created in his membrane by some dinitrophenol. Rotenone and cyanide stop my electron flow and halt the calculation of my P2O ratio. Both rotenone and cyanide stop his electron flow and halt the calculation of his P2O ratio. We'll talk more about some of those things next week.